Okay, please let me know if that is not showing the presentation slide. All right. Thanks everybody for joining again. For those who just joined, just to give a little bit of background on where this work is coming from. This was a year-long study that I led for the last year while I was at the Wild Salmon Center. That was a project that was funded by NIFWIF and U.S. Fish and Wildlife Service with the goal of pulling together data on Chinook salmon throughout the whole Klamath Basin to try to find new understandings and synthesize information across the entire basin. So this is a project that I led with a really large group of folks, of people from diverse knowledge backgrounds and diverse entities that help provide a lot of input on this project. And again, this was a project that was funded by Fish and Wildlife Service with the intention of pulling together a lot of the information on Chinook salmon in the basin. So a project like this that is trying to synthesize information that already exists wouldn't be possible without all the folks that have been involved in collecting the data. And so this is all the different groups in which we used their data to do this project. And I know a lot of people that are here have work for these different groups and are part of collecting the information that I used in the analysis. So thank you for your involvement in making this work possible. So we're here today, or this project came about to focus on Chinook salmon abundances over time. And their abundances in the last decade have been in a prolonged period of historically low abundances. And this period of low abundances really began over a decade ago during the California mega drought that happened from 2012 to 2015. And then the ocean heat wave, or the blob, that happened almost around the same period of time that really impacted Chinook salmon in this basin. And so since these impacts, the population has been at really low abundances from 2015 to 2017 that led to the stock being designated as overfished in 2018. And since then, we haven't seen this population meet the threshold of being rebuilt. And actually, in the last 8 of the last 10 years, the escapements for Chinook salmon in Klamath Fall Chinook have fallen below the target 8 of the last 10 years. And I want to emphasize that the low abundances that we've seen in the last decade have occurred in the past, such as like 2004 and 2005. But what really makes the current phase unique is how long this period of low abundances have been, how prolonged it's been. In the past, we've seen really positive environmental events lead to a recovery of the population abundances, but we really haven't seen that in this current state that we're in. And so one of the goals of this project was to understand why environmentally we haven't seen a rebounding in the population abundances. So that kind of leads to how this project came about, was to look at different environmental factors that have affected Chinook salmon throughout their life cycle in the basin. Another goal that was identified to us by Fish and Wildlife Service is to pull together information on populations in all the different tributaries and to understand how populations in the different tributaries are affected by the environment in different ways, and then also to identify some key recovery focus in the basin. So that brings me to the approach that we took. We took a whole life cycle approach in this analysis, and we broke it down into four different analyses and life stage pieces. And so the first part of the analysis was looking at the production in the tributaries. So looking at the adults coming back and spawning and the juveniles rearing in the tributaries. And then once juveniles move from the tributaries into the mainstem, what is their outmigration like in the last few decades through the mainstem? And then once these fish go from the mainstem to the ocean in their first year in the ocean, what environmental factors have affected their ocean survival, and what is ocean survival like in their first year? And then lastly, once these adults reach harvestable size, how have they been impacted by fisheries in the last few decades? So I'm gonna dive right in and start with the first piece of the analysis, which is looking at freshwater productivity in the tributaries. And so the data that we were able to compile looking here is information on adults coming back through the different survey methods from the different agencies, and then the subsequent data on juveniles leaving the different tributaries from the screw trap data. So for this piece of the analysis, we were able to look at the screw trap data in a lot of parts of the basin. We had information on 3 screw traps in the mainstem, screw traps on 5 of the different tributaries in the basin, and then also screw traps on the upper and lower Trinity River. And for this piece of the analysis, we were looking at data from the last 20 or so years, because that's when a lot of the screw trap data and monitoring began. So this here is basically showing the juvenile abundance relative to the amount of spawners upstream of each of these different screw trap monitoring sites. And so you can see that for the mainstem traps, we have a decline in the amount of juveniles that we are seeing per spawner as we move further downstream. The sites with the highest productivity, with the highest amount of abundance per spawner, were the uppermost mainstem monitoring site, Shasta River and Camp Creek were some of the highest abundance per spawner. And then the sites with the lowest juvenile abundance per spawner were the 2 screw traps on the Trinity River. So when we look at the biomass, so accounting for the size of the fish in all of these different monitoring sites, we see that the 3 monitoring stations along the mainstem river actually become quite equal once you account for the fact that fish are larger as they move down, but there might be fewer fish from mortality that happens along the way during the migration period. And then when we also account for the biomass of fish, we see that the Shasta River clearly has a higher productivity compared to the other monitoring stations, which is what a lot of people in the basin also assume, with the Shasta River being a highly productive system. The site with the lowest productivity in terms of biomass per spawner was the uppermost Trinity River site at Pear Tree, and this monitoring station is right below the Lewiston Dam there. This is showing the juvenile biomass trends over time in all the different monitoring stations. And what I was expecting here, because of the low productivity we've seen in the last decade, was potentially a decline in productivity over time. But actually what we see is, for the most part, pretty steady juvenile biomass per spawner across years. And if anything, we see in some of these sites an increase in biomass per spawner. And I think partially why this is the case is because there's lower abundances, there's less density dependence. So as a rate, the biomass per spawner has been higher in more recent years because of the low abundances. What's also really interesting was that when we looked at correlation between sites and whether there was high correlation, we saw really low rates of correlation between the juvenile monitoring stations, which is overall a good thing when we're trying to think of different tributaries having asynchronous dynamics and not highly covarying with each other. So for this piece of analysis, we also looked at what environmental conditions affected the productivity in each of these tributaries. And some of the different effects that we were interested in looking at were number of spawners, whether there was an effect of density dependence on productivity. We also wanted to look at conditions during the spawning and the incubation period. So we looked at whether spawning flows, the average spawning flows, which was a proxy for when winter flows began in the fall, had an effect on productivity. We also looked at whether temperatures during the spawning and incubation period affected productivity, and then peak flows and then average flows during this period too. And then once juveniles emerged from these different tributaries, was there also an effect on the temperatures and the flows during the springtime on the juvenile productivity? So this is going to slowly walk through the different results for the different juvenile monitoring stations. And so when we evaluated spawner abundance on total productivity, so biomass per spawner, we saw a negative effect of spawning abundance. So this is signaling density dependence that was happening at all the different juvenile monitoring stations. When we looked at spawning flow in all the different sites, we only found a weak negative effect on the Shasta River. And so here, when I'm walking through the results, if there's a symbol, that means there was a significance to the 80% confidence. And then if there's a symbol and there's the shading, that means it was significant to the 95th percent confidence. For spawning and incubation temperatures during the fall, we saw a negative effect of temperatures at the uppermost Trinity River site at Pear Tree. So warmer temperatures in the wintertime led to lower overall biomass productivity. We didn't see an effect of spawning flows at any of the monitoring stations during the wintertime, which overall seems to suggest that the flows at all the different tributaries are sufficient during the wintertime, at least for the incubation period. And then for peak flows, we saw some negative effects of peak flows at the Willow Creek and at the uppermost Klamath River site at Bogus Confluence. And talking to folks that work in these regions, overall, it doesn't seem like there's a lot of scour that happens near these sites. And so it might be some other effect of high flow event that negatively affects juvenile productivity. And then looking at springtime flows, outmigration flows, we see really a blend of positive and negative effects at the different monitoring stations of flow. And I think for the most part, what is happening here is a mixture of biological signals and then also effects of flow on sampling. And so if there's high flow, that could lead to, fish leaving earlier. It could also result in fish staying in the river and leaving after the screw traps are removed. And so it could be that flow is having different effects on the measured productivity because of both sampling and biology. And then lastly, outmigration temperatures. Interestingly, we saw an opposite effect at the uppermost versus the lowermost Trinity River site. So we saw a positive effect of springtime temperatures at Pear Tree on biomass productivity, but a negative effect of temperature further downstream. And overall, when we compare the different juvenile monitoring stations, we see that there's sometimes conflicting effects of these environmental variables on productivity. So for example, on Trinity, we have the positive effect upstream and the negative effect downstream. And then on the Klamath River, also a negative effect on the uppermost sites, but then a positive effect of flow further downstream. And I think what this is capturing is that these traps are monitoring different life stages. So, for example, These traps are monitoring the fish at different life stages. So further upstream, they're primarily catching fish as fries. So the uppermost sites are going to be monitoring survival and during the egg and the fry ring stages, whereas traps further downstream are not only capturing the incubation and the fry rearing, but they're also capturing the variation in outmigration survival as well. Looking through the results, we saw that the Shasta River had the highest productivity across all stations. It had very high abundance per spawner, but also the highest biomass per spawner. And this is partially in a lot attributed to the fact that the Shasta River is a groundwater dominated system. And so the springs in the Shasta River result in the temperatures in the wintertime to be warmer than rainfall. But then the springs in the springtime also cool down the river and the river is cooler than a lot of neighboring rivers during the springtime. The groundwater also increases the base flow during the fall, which allows Chinook salmon to spawn and not earlier in the year and not necessarily have to wait until the fall rains begin. And so all of these factors together help give Chinook salmon in the Shasta River advantage because the adults are able to come back earlier. With the warmer temperatures, the eggs are able to incubate faster. And then with the high productivity, those fish are able to grow and leave the river at an earlier and larger size. What's interesting is that in the Trinity River, superficially, we have a really similar pattern in the Trinity River. We have warmer temperatures in the winter and cooler temperatures in the summer, and then also higher base flows. But we saw the opposite effect or the opposite pattern happening in the Trinity River. We saw very low biomass productivity. And this partially speaks to the fact that the warmer temperatures and the cooler temperatures in the springtime are from the dam that's regulating stream flow artificially. And so even though it's warmer in the winter and cooler in the summer, it's happening from a single point source versus happening throughout the basin. And it doesn't look the same as how the springs are affecting water temperatures in the Shasta River. At the pear tree site, we saw a negative effect of temperatures in the wintertime. And this is partially because the water temperature in the Trinity River is already warmer than typical because of the water coming from the dam. And so even warmer temperatures in the wintertime further deviate the water temperatures from what's natural. And then in the springtime, with the waters coming from the dam being cooler than what would happen naturally, colder weather in the spring further deviate the temperatures from the natural temperature regime in the Trinity River. You also see in the results that the minor tributaries had pretty high productivity, and it might be that these tributaries are highly productive, but it could also be that these tributaries have higher sampling efficiencies because they're smaller, and so they're easier to have a higher trapping efficiency and capture and more accurately estimate the abundance of fish leaving the creeks. So, in summary, for this portion of the analysis, some of the key take-home points is that the groundwater-dominated system in the Shasta River led to a really high migrant productivity in the Shasta. The artificial flows in the Trinity River that are also altering the thermal regime in the river, similar to the Shasta River, but in a different way, may be inhibiting the productivity in the Trinity River. We also saw that there was an effect of density dependence at all the sites. So there is capacity limitations in all of the different basins that were monitored. And overall, surprisingly, we didn't see a declining trend in productivity in the time of our study period. And then there was low synchrony when we evaluated the correlation between these different sites across the tributaries, which is overall a good thing when we think of trying to have a diverse population dynamic in the basin for greater stability overall. So next, I'll move on to the next portion of the analysis, which is looking at the downstream migration of fish. And so for this piece of the analysis, we were working with the stream salmonid simulator model, which is the S3 simulations. And this model simulates the movement of fish downstream. And as they move downstream, it also simulates their survival rates and also their growth rate based on the bioenergetic conditions in the river. What's also really important about the S3 model is that there's also a disease component to this model that allows us to estimate the impact of sea shasta mortality, which is a disease that is primarily concentrated in this orange region right here. And so the model is able to simulate, based on the amount of spores in the water and the temperature, how much are fish passing through this range getting infected by sea shasta and dying along the way before they reach the river. So some of the questions that we were interested in exploring with the S3 model is looking at what the outmigration survival rate is for each of the source populations. When does each of the populations reach the ocean and at what size? And then with the growth bioenergetic piece, what is the growth potential in the mainstem over space and time? And then how are each of the populations affected by sea shasta? So this is basically showing the average survival of the different source populations in the mainstem over time. So when you look at the survival rates, you see there's a really big range of estimated mainstem survival. The differences are really attributed to location in the basin. So basins, subbasins further upstream, such as Bogus Creek or or fish that are in the mainstem, have very low outmigration survival rate because they have to travel the greatest distance, whereas fish further downstream, such as fish entering the river from the Trinity River, have a higher survival rate because they have to travel a shorter distance. We also see a really big effect of size. So fish that are released from Iron Gate hatchery at different times of year and at different sizes have a dramatically different estimate of survival. What's interesting is that in all the different source populations, we saw a pretty clear decline in the estimated survival rate of hatchery fish, both in fingerlings and yearlings. And for both of these, it's partially because fish have been released at smaller sizes in more recent years. And so in the model, fish that are smaller spend more time traveling through the river and are more likely to die before reaching the ocean. But there's also, for the fingerlings that are released in the early summer, a big effect of disease on mortality too. So from our S3 simulations, we estimated the impact of sea shasta on the different source populations. And so in more recent years, we've seen more frequent years with sea shasta mortality and also greater intensity in the estimated mortality rate too. I also want to point out here in this figure that in the light blue here is fish from the Shasta River, which actually these fish have to migrate through the whole region in which the sea shasta spores exist, but they experience much lower sea shasta mortality, we estimate, than the other populations, including populations further downstream, such as the Scott River, which actually these fish have to migrate a shorter distance within the area in which sea shasta spores exist. But because of the timing in which fish from the Shasta River are in the mainstem, their mortality rate is estimated to be lower. So fish from the Shasta River are primarily in the area in which the sea shasta spores are during the early parts of the summer, where the spore concentrations are a lot lower and the temperatures are also a lot lower, and they're also larger, so they're moving through this area more quickly and have a lower rate of being infected and a lower rate of dying from sea shasta versus the other populations that have to swim through that area too. This figure here is showing growth potential throughout the different parts of the basin. So in each figure, I show the growth potential over time across the 20-year period. And then for each of the columns, we have the growth potential in the start on the first day of every month. And then for the rows here are different areas of the river. So this is river kilometer on the right. So the lower part of the river is on top and the upper part of the river is on this bottom row here. And overall, when we look at this figure, we see that the growth potential in May has been increasing over time. And this is partially because the temperatures in May are normally pretty cool. And so as temperatures get warmer with climate change, this is moving the growth potential closer to the thermal optimum. With temperatures getting warmer, however, we see during the summertime when it is already pretty warm that even warmer temperatures are moving temperatures away from the thermal optimum. And so the growth potential has really declined for the months of July and August over time. This figure here is showing the estimated ocean entry size and date of fish from the different tributaries. So overall, basin-wide, the average ocean entry estimated size was around 95 millimeters, and that the date occurred primarily around the late June period. In the top right here in the light blue, we have fish from the Shasta River. So fish from the Shasta River are estimated to arrive to the ocean at a much bigger size and also at an earlier date. On the left here in the smaller sizes range, we have fish from the Trinity River and also Trinity River hatchery. Overall are arriving at a smaller size and a lot of times at a later date. So this goes back into the conditions of the tributaries that's affecting the size and timing in which fish are reaching the ocean. So in the Shasta River, we already mentioned that with the groundwater and springs that are in the river, that the adults are able to come back earlier, and that the fish also are able to grow more quickly, so they're able to move into the main stem at an earlier and at a larger size and avoid a lot of the disease. And then also in more recent years, with the growth conditions improving earlier in the summer, their timing in which they're in the river actually now better overlap with the peak growth conditions in the main stem. This is the opposite, or very in contrast to what we see in the Trinity River. The temperatures in the Trinity River are artificially much cooler than in other parts of the basin, and so growth rates in the basin of the Trinity River might be a lot slower, which may be inhibiting the size of these fish, and so they might be holding in the river for a longer period of time and moving into the main stem at a later date and entering the ocean later and at a smaller size. So when we looked at trends in the ocean entry size for the different source populations over time, we see there's a decline in the ocean entry size for a lot of the different populations here, for the hatchery and for some of the natural populations too. And the main reason why we see a decline in the size in which fish are entering the ocean is because of the day in which they're arriving to the ocean. So in a lot of these populations, we also see an earlier ocean entry arrival here. So on the axes here, I have the day in which they arrive to the ocean. So the upward, if you see an upwards trend, that's showing that fish are on average arriving to the ocean earlier. So both these two figures together tell a story that these fish are arriving to the ocean earlier and at a smaller size in more recent years. And it's not so much in the model that fish are moving out more quickly or they're migrating faster, although that could be happening. The reason why we see this in the model is because of the disease that's been happening later in the year that's resulting in mortality in the later migrants. So the window of time in which fish are arriving in more recent years has been truncated. And so there's been an overall shift in the average time to earlier and smaller sizes because the later migrating fish are not surviving to the ocean. And this has huge impacts on ocean survival, as we can imagine, with fish arriving at a smaller size. So in summary, for this portion of the analysis, looking at juveniles in the mainstem Klamath River, overall we see that the suitable conditions in the river, both in terms of disease and growth potential, have been shifting to earlier in the season. And as it gets earlier in the season, the time in which fish have to rear in the river is becoming shorter as well. There's also a great disparity in more recent years between the early and later migrating fish, with the earlier fish benefiting from the warmer temperatures in the springtime, and the later fish that are later fish having higher mortality and experiencing temperatures that are way past the thermal optimum. So now moving into the third piece of the analysis, which is looking at ocean survival in the first year. So when these fish enter the ocean, we believe they experience a really high period of mortality in the first few months in which they enter the ocean, mostly from predation right when they enter the ocean. And so size when they enter the ocean and growth in the first few months are going to be potentially really important for determining their survival when they're in the ocean. We also believe that there may be periods in the wintertime that could be a period of really high mortality because of limited food that's available. And so it's also possible that during the wintertime, they also may be dying because they aren't metabolically able to survive in the wintertime. So how did we estimate survival in the first year of life in the ocean? Because understanding ocean survival can be a lot more difficult with limited data available. So to look at the first year ocean survival rate, we conducted cohort reconstructions for all the different years of fish heading out to the ocean. And so we have information on the adults coming that are harvested and the adults that are coming back. So information on adults of different ages coming back to spawn or harvested in the river, and then also information on fish that are harvested by ocean fisheries as well. And so the cohort reconstruction essentially pieced all the different fates of fish over time together, and then also accounting for natural mortality that could be happening, and back calculating to estimate how many subadults there were in the ocean to begin with. And so this allows us to estimate the first year ocean survival based on the number of juveniles that went out to the ocean and the reconstructed number of age 2 fish at the start of the subadult period. So this is what our estimates of first year ocean survival look like for fish in the basin. So you can see that there's a lot of variation from year to year in ocean survival rates, and that generally it ranges from 2 to 3% to about 10% in their first year. And we also have information on the ocean survival rates for Redwood Creek, which is the basin right south of the Klamath River Basin. And our estimates independently line up really well, both in terms of absolute numbers and also year-to-year correlations. So overall, that's a good indicator about the accuracy of these estimates. So our question here with the ocean survival is what conditions in the are there any conditions in the ocean that we can look at to try to better predict ocean survival? So in terms of looking at predictors for ocean survival, some of the factors that we looked at were ocean temperatures. So whether average ocean temperatures off of the mouth of the Klamath River predicted survival. We also looked at temperatures that are within the suitable habitat or within the suitable temperature range off of the coast of the Klamath River. We also looked at an upwelling index, so the beauty index to see if there's an prediction or if the upwelling index can predict survival. And then lastly, we're lucky enough in this basin to have data on oceanographic other oceanographic data from Trinidad on the Coral Sea. And so we also included information on krill densities and krill sizes, whether any of the food could also be used as a predictor for survival. So we looked at whether the temperature, upwelling, and the food size and density during the summer and the winter time were good predictors for survival. And then we also looked at the average smolt size and the timing to see whether the cohort characteristics also predicted survival too. And overall, in our model selection process, we found that winter temperatures and winter food densities were the strongest predictors for ocean survival for the first year survival in the ocean. Summer zooplankton size also came out in a top model for predicting ocean survival, but again, the winter conditions were the strongest predictor for ocean survival. And this was initially surprising for me, but after thinking about what these results mean, it made a lot more sense because originally I thought the summer conditions are understood to be really important for the growth and the survival of these fish, but really positive growth conditions in the summer might not necessarily mean high survival rates. If the ocean is really productive and fish are growing really well, there might also be a lot more predators, a lot more competition. So I think the effects of high productivity in the summer are a lot more complicated in how they affect survival. Compared to the wintertime, if the wintertime is really about metabolism and bioenergetically able to survive the wintertime, then temperatures are going to be really important for lowering the metabolism of fish, and any food in the wintertime is also going to be important for supporting the fish through this winter period. Once these fish grow to be a harvestable size, they can be harvested by different fisheries. And so we also looked at the impact of ocean fisheries on Chinook salmon. So this is showing both recreational and commercial fisheries in the ocean on the different age classes of fish. And so you can see that as fish get older, they also get bigger. And so they're more likely to be of harvestable size as they become larger. And we also see that since the stock became designated as overfished in 2018 because of low abundances, the impact rate from the ocean fisheries really has not gone down since 2018 up until the fisheries closure. And in fact, for some of these impact rates, we see that they've been higher after the overfished designation than before. So why were the impact rates so high during this period when the stock was already designated as overfished? Two of the main reasons is that the forecasting models for Klamath salmon abundances overestimated how many fish there were in the ocean, and the harvest models that estimated how many fish people would harvest based on the management regulations, underestimated and how many fish would be harvested within the season. So both of these factors together and in different years led to a higher impact rate than intended during this period. As once fish begin maturing and moving into the river, they can also be harvested by in-river recreational fisheries and also tribal fisheries. And so here we have the impact rate at the different ages by both of these fisheries. And you can see that it reflects pretty strongly on the regulations and the gear types of these fisheries. So for the recreational fisheries, we see that age 2 fish have the highest impact. And this is because in a lot of years, the regulations in the recreational fisheries is allowing to keep one jack and one adult. So there's a lot more jacks that are being caught relative to all the different adult age classes. And then for the tribal fisheries, a lot of the gear is using nets. And so the nets are can be pretty size selective. And so we see a pretty strong effect of size on the age impact rates. So all of these different fisheries have cumulative impacts on salmon abundances. So fish that survive to spawn has to escape all 4 of these different fishery types. And so this figure here is showing the total impact of fisheries from the ocean, commercial, and recreational fisheries, and the river, recreational, and tribal fisheries on spawning abundances. And so we simulated how many, if without these fisheries and based on the life history of these fish, how many adults would have came back if there wasn't fisheries. And this reflects the proportion in which the actual number of adults came back relative to the amount that would have potentially come back without fisheries. So to summarize this fishery portion, the fisheries impact during the last 10 years have had a significant impact on the adult returns in the river, and especially during the period since the overfished designation before the fisheries closure, the impact remained relatively consistent or even increased in the ocean during this overfished designation. And again, this is partially due to the abundance forecast models overestimating how many fish there were, and the harvest models underestimating how many fish would be harvested, and the harvest goals being exceeded in some of the years of the fisheries. So looking forward to addressing these scientific and management errors, it's really important to understand the population dynamics and adjust the fishery forecast models and also the harvest models in times of error. And the Pacific Fishery Management Council recently has changed the population dynamic model to reflect more recent years of data, but continuing to understand the population dynamics and improving these models are really important for the intention behind the management to be achieved. And then also considering a buffer, both explicitly in science and forecast models, and then also holistically by managers are really important when the population is already very close to or below the target to try to avoid periods of even more prolonged periods of low abundances. And then going back to the freshwater conditions, we also saw that the mainstem conditions in the summer have been very poor in the last decade, and that this has affected nearly all the different populations in the basin. We saw that the Shasta River was one exception to this, because of the fact that they moved through the mainstem earlier in the year compared to the other populations. The poor mainstem conditions really have a disparate effect on different life histories. So it's really impacting those later emigrating fish that are in the river later in the summer. And because of the high mortality of those later emigrating fish, we saw a shift in the average ocean entry timing to those earlier and smaller individuals, which we are which we think overall would have lower survival on average than larger juveniles. So in terms of how these conclusions can help shape strategy, it's really important to support both those early migrating fish that are leaving their natal habitats earlier in the year and moving downstream, and also the fish that are moving through the river in the summertime. So for the early migrating fish that are moving downstream, they need habitats further downstream, such as the estuary and non-natal tributaries in the lower river to support their growth before they go out to the ocean. And for fish that are moving later in the summer, cold water refugia is going to be really important for them to survive during their outmigration period. So just some additional conclusions that I have from working on this project. Looking at the data over the last 20 years, we saw that in the last 10 years that the environmental conditions for salmon in the Klamath Basin have generally been poor to average. And in the periods and the earlier part of the study, we saw some positive freshwater and ocean conditions that helped recover and rebound the population from low abundances. And we really didn't see that in the last decade of our study. And this is only going to continue more and more with climate change, and that we should not expect good conditions in the future to help rebound the population. We need to manage the population with future climate conditions in mind and expect in the future to have fewer good years to help rebound the population and more years that will lead to high mortality. So I'll just close out again by thanking all of the different entities and the people that work in these different groups that contributed data to this project that really made a project like this possible. So thank you again. And if there's any time, I'm happy to answer questions. And there's also my email here in the top right corner if you want to reach out to me individually and talk about things further. So let me go ahead and stop the recording.